Hello friends, I am going to discuss the organization of human genome. The human genome is a complete set of nucleic acid sequences encoded as DNA within the 23 chromosome pairs in cell nuclei and also a small amount of DNA which is found in mitochondria. The human genome is divided into 46 DNA molecules or chromosomes which consists of 22 pairs of autosomes and 2 sex chromosomes. Together, these molecules contain over 3 billion base pairs of DNA. It stands to reason that human genome must be extensively packaged in order to fit inside the nucleus, the size of which is in micrometer range. The human genome includes the coding region of DNA, which encodes all the genes between 20,000 to 25,000. However, the estimate of the number of human genes has been repeatedly revised with the advancement in genome sequencing and gene finding methods. Besides the coding region, the human genome also contains the regions which do not encode any genes. So only a fraction of genome is occupied by protein coding exons, and the majority of non-exonic sequences consist of repetitive or transposable elements. The functional exons contribute merely 2% of a genome. Up to 50% of genome is occupied by repetitive elements. The remaining 48%, called the unique DNA, most of which probably originated in mobile elements which diverged over time beyond recognition and for which as yet no function has been elucidated. The DNA sequence of the genes consists of exons and introns. The exons represent the portion of the gene that encodes for amino acids, and the introns represent the intervening sequences between the exons. The intronic sequences do not contribute to protein specifications, rather they are spliced out during the process of transcription. Chromosomes appear of nowhere at the beginning of mitosis during cell division and disappear once again when the cell division has ended. In non-dividing eukaryotic cells which are said to be in G0 phase and those in interphase, the chromosomal material is known as chromatin, which is amorphous and seems to be randomly dispersed in certain parts of the nucleus. So the physiological state of genomic DNA is in the form of chromatin, where it is bound to histone and non-histone proteins. Chromatin fibers consist of protein and DNA in almost equal proportions. The DNA in the chromatin is very tightly associated with histone proteins, which package and order the DNA into structural units called the nucleosomes. Histones are by far the most abundant proteins in chromatin, which are basic proteins rich in basic amino acids like lysine and arginine. Histone proteins are also evolutionarily conserved proteins, which means that they are very similar across vastly different species, of which H3 and H4 being highly conserved ones. The nucleosome is the basic structural unit of chromatin, which consists of a histone octacore comprising of two copies each of H2A, H2B, H3 and H4 histones. Histones, particularly their amino and carboxy terminal tails, are subject to post-translational modifications like acetylation, methylation, phosphorylation, ubiquitinylation, which have a vast effect on their structural and functional properties of chromatin. Now, as you can see in the diagram how chromatin is organized, the double helical DNA, which is represented by the two strands joined together through base pairs, is 2 nanometers in width. The DNA then gets wrapped around histone proteins, which in turn forms a nucleosome, and DNA gets further compacted. A DNA molecule which gets wrapped around nucleosome core of 10 nanometer diameter is the lowest level of chromatin organization. Chromatin in cells, however, exists as tiny dots of 30 nanometer diameter. The wrapping of DNA around nucleosomes represents the first level in packaging, which effectively shortens the length of chromosome by 7-fold. DNA molecules interact with histone proteins as can be seen in the figure, to form the nucleosomes. A histone octamer interacts with DNA to form a beads on a string structure. The H1 histone links two nucleosomes together and the nucleosome swirl around itself and gives rise to chromatin fiber, which is 30 nanometers in diameter. The spacing of the nucleosome beads provide a repeating unit of about 200 base pairs of DNA, of which 147 base pairs are bound tightly around the histone octacore, and the remainder serves as a linker DNA. The linker DNA is bound by H1 histone proteins. This is confirmed by the brief treatment of the chromatin with enzyme that digests DNA, like micrococcal nuclease. If we look at the individual nucleosome, it can be seen that it consists of 5 different types of histone proteins. The main body is an octamer, which is formed from 2 copies each of H2A, H2B, H3 and H4 histone proteins. The DNA is wrapped around the histone octacore in the form of left-handed solenoidal supercoil. The fifth histone protein, called the H1 histone or the linker histone, links the two nucleosomes together, which covers 30 to 50 base pairs of DNA. Now, as can be seen in the figure, how chromatin gets organized. The DNA double helix, which is 2 nanometer in width. As we go along, there is a DNA which gets wrapped around histone proteins, giving rise to a nucleosomal structure which appears as a beads on a string, which compacts the DNA up to 7-fold and gives rise to chromatin, which is 10 nanometers in diameter. Next, the nucleosomes are swirled around itself and gives rise to chromatin fiber, which is 30 nanometer in diameter. This chromatin is then looped and further wound and packaged to give rise to 300 nanometer structure, which then finally gives rise to a highly ordered compacted structure known as metaphase chromosome. Thus, the end result is that the DNA is tightly packed into the structure known as chromosome. It is important to realize that chromosomes are formed only when cells are dividing. As mentioned earlier, the DNA in cells exists as chromatin fibers of 30 nanometer diameter.